Hello everyone, my name is Viktor Krivenkov. I am a postdoctoral researcher in the Laboratory of Hybrid Photonic Nanomaterials of the National Research Nuclear University, Moscow Engineering Physics Institute in Moscow, Russia. Our studies are dedicated to the design and fabrication of nanomaterials working in the regime of light-matter coupling, even in the non-linear regimes. Here I present you the summary of our recent results about the enhancement of the photoluminescence of semiconductor quantum dots coupled with micro and nano cavities. Semiconductor quantum dots are spherical fluorescent nanocrystals which are known for their unique optical properties. Due to the small diameter, quantum effects are very strong. This leads to a discrete structure of their energy levels and a strong dependence of their absorption and luminescence spectra on the nanocrystal diameter. In practice, quantum dots are attractive for a wide absorption spectrum not limited to the blue side of wavelength, large values of the absorption cross-sections, and for a narrow luminescence spectrum with a quantum yield that can reach 100%. All that makes quantum dots a promising material for the fabrication of novel nano-hybrid materials for photovoltaics and optoelectronics. One of the most impressive features of quantum dots is their capability for highly efficient two-photon absorption. Two-photon absorption makes it possible to utilize the energy of photons, the energy of which is less than the band gap. Moreover, this process is non-linear in intensity, which determines some of its features, namely the need to use a high excitation intensity and the possibility of three-dimensional localization of excitation by excitation focusing. Indeed, in a laser beam focused by a lens, the highest intensity will be concentrated in the region of the beam waist, and it is there that the highest probability of absorption will be observed. The possibility of three-dimensional localization of two-photon absorption makes this process promising for medicine. In the case of quantum dots, which are used in the field of biomedicine, this also gives us the possibility of using infrared wavelength for biomarkers excitation. Indeed, the so-called transparency windows of biological tissues are located in the near-infrared wavelength range. The use of excitation in the spectral range of these windows allows radiation to penetrate into biological tissues deeper than the other spectral ranges. For this application, quantum dots are significantly better than traditional organic dyes due to the uniquely large two-photon absorption cross-sections of quantum dots. This allows to increase the depth of the detection inside biological tissues further. All that together creates the prospect of using two-photon excitation of highly luminescent semiconductor quantum dots both for ultra-high contrast biovisualization and for fabrication of and design of nano-biohybrid upconversion solar cells and nonlinear optical photodetectors. As I mentioned before, the two-photon absorption quantum dots requires the use of high intensity of laser excitation. However, this is not always possible due to the limitations associated with the absorption of radiation by the surrounding medium, especially in the case of bioimaging. Therefore, additional measures are needed to increase the photoluminescence intensity of quantum dots under two-photon excitation. The first way to increase the photoluminescence intensity is to increase the quantum yield of quantum dots. Indeed, when interacting with molecules of the environment, especially in the case of bioimaging, the quantum yield of photoluminescence of quantum dots usually decreases several times due to the appearance of additional non-radiative excitation relaxation channels. In order to increase the photoluminescence of quantum yield, it is necessary to increase the rate of radiative relaxation. This can be done by using the Purcell effect. The nature of this effect is an increase in the density of photon states near the fluor at the wavelength of its photoluminescence, which leads to a significant increase in the rate of radiative relaxation. One of the most promising methods for its implementation is the fabrication of microcavities based on porous silicon photonic crystals. The second way to increase the photoluminescence signal of quantum dots under two-photon excitation is to enhance the local field near metal plasmonic nanoparticles. Indeed, plasmonic nanoparticles can serve as open nanocavities, then can concentrate the electromagnetic field on the nanometer scale. This makes it possible to significantly increase the local intensity of the excitation field and thus significantly increase the probability of absorption for quantum dots located nearby. In this presentation, I'll discuss of both ways. For experiments, we use semiconductor quantum dots with multi-layer shell. Quantum dots consist of cadmium selenide core and 3 monorays of zinc sulfide, cadmium sulfide, and finally zinc sulfide. The diameter of used quantum dots was about 4.5 nanometers. The maximum of the photoluminescence spectrum was at 560 nanometers, with full width at half maximum of 35 to 40 nanometers. The photoluminescence quantum yield of used quantum dots was 90% in the hexane solution, and corresponding photoluminescence lifetime was 30 nanoseconds. In some experiments we used gold nanorods. They have a diameter of 12 nanometers and length of about 50 nanometers. On the graph one can see the plasma spectrum of the gold nanorods. The longitudinal plasma mode is positioned at the spectral maximum of 800 nanometers, which corresponds to the near-infrared transparency window of biological tissues. From the left top graph one can see that in this spectral region there are no transitions for the quantum dots absorption. Fabrication of porous silicon photonic crystals was performed using the procedure of the electrochemical etching of monocrystalline p-type silicon. Etching was performed by means of a self-made etching setup. The mixture of hydrofluoric acid and ethanol was used as an etching solution. In this study we have fabricated two samples, distributed Bragg reflector and microcavity. Simple reflector was formed by stacking 25 pairs of layers with high porosity of 75% and low porosity of 58%. Porous silicon microcavities were formed by stacking the front and rear mirrors and the half-wave cavity layer which was placed between them. The front and rear mirrors included 5 and 20 pairs of layers, respectively. Note that such type of porous silicon does not demonstrate inherent photoluminescent properties. After etching, samples were thermally oxidized to prevent the strong charge transfer from quantum dots to the silicon surface. Finally, oxidized samples were treated with surfactants in order to make their surface hydrophobic and improve the infiltration of nanocrystals. Embedding of quantum dots into the porous silicon substrate was performed by drop casting of the hexane solution of quantum dots. The photonic band gap of the microcavity was about 150 nanometers wide, with its agent mode concentrated at 580 nanometers, and its quality factor was about 83. This microcavity design was chosen in order to provide partial matching between the quantum dots emission maximum and the microcavity agent mode, and to suppress the rest of the inhomogeneously broadened photoluminescence, which remained uncoupled. To investigate the effect of gold nanorods on the two-photon absorption of quantum dots, we have fabricated two thin-film hybrid structures consisting of quantum dots in a 20 nanometers thick PMMA layer using the spin coating technique. We chose this thickness because it had been previously shown to be within the optimum range for the maximum photoluminescence enhancement. After fabrication of the initial 20 nanometers quantum dots in PMMA layers, one of two samples was covered with gold nanorods by dropping from aqueous solution and drying the samples, which resulted in the formation of the disordered surface films. At first we measured the photoluminescence properties of quantum dots in the porous silicon microcavity. One can see that after embedding of quantum dots into the microcavity, their spectra split into two peaks. The first one at 575 nanometers with width of about 50 nanometers corresponds to the wavelength of the microcavity aging mode and may be related with the effect of weak coupling of quantum dots with the aging mode. The second peak at 550 has the same width as the initial ensemble quantum dots in the solution and can be attributed to the uncoupled part of the ensemble. In bottom figure we have plotted the dependencies of peak intensities on the pump power in order to indirectly demonstrate that these two peaks have different radiative relaxation rates. As we increase the pump power, the initially quadratic dependencies saturate and the saturation thresholds was higher for the long wave peak. We approximated the experimental dependencies with the presented equation and found that the observed difference in the saturation thresholds can be only a result of the significant change in the radiative lifetime. Quantitative estimation of the Purcell factor could be made by comparing the radiative relaxation times extracted from the photoluminescence kinetics of the quantum dots coupled to the microcavity aging mode and the uncoupled quantum dots ensemble. In order to exclude the influence of the non-radiative relaxation pathways arising from the interaction of quantum dots with the surface of porous silicon, the control experiment has been made. As control samples, we used quantum dots embedded inside the single black reflector and quantum dots dispersed in the polymer matrix on a glass substrate. The total radiative and non-radiative relaxation rates were calculated. Using the final values, we can estimate the Purcell factor or the radiative rate amplification factor as 4.3. The alternative approach for the increasing of the two-photon-excited photoluminescence is to increase the two-photon absorption probability. Two-photon absorption cross-section is the main parameter that reflects the two-photon absorption efficiency, regardless of the amplitude of the excitation field. Previously, we have shown that the photoluminescence saturation measurements allow finding the two-photon absorption cross-sections of quantum dots in the ensembles without prior knowledge of particle concentration and quantum yield. To describe the saturation, we use the equation that expresses this process as a function of the excitation intensity. In the experiment, we measured the dependence of the photoluminescence intensity on the excitation intensity for two samples: first quantum dots in the polymer matrix, black squares on the graph, and quantum dots in polymer matrix covered with gold nanorods, red circles on the graph. We fitted these dependencies using the presented equation and found that the highlighted part of the equation was different for these two samples. Assuming that the laser lifetime was the same in both cases, the change is related either to the change in the excitation intensity or to the absorption cross-section. If we use only the far-field excitation intensity, then we need to use the changed effective two-photon absorption cross-section, which change reflects the change in the position of the deflection of the saturation dependence. Thus, we have extracted the two-photon absorption cross-sections from these dependencies on the different excitation wavelength. One can see that the effective cross-sections near the gold nanorods were much higher than the simple cross-sections of quantum dots. Using these values, we have calculated the enhancement of the two-photon absorption, which was up to 12. One can see that the calculated increase of the excitation intensity follows the plasmon spectrum of the gold nanorods, which prove that this enhancement is the result of the plasmon-induced effect. In conclusion, we have investigated the two-photon excitation of quantum dots whose energy transitions are weakly coupled to the aging mode of either porous silicon microcavity or to the plasmon mode of the gold nanorods. We found that inside the porous silicon microcavity, the radiative rate of the two-photon excited quantum dots accelerated by a factor of 4.3, and the two-photon absorption efficiency was increased by a factor of up to 20 near the gold nanorods. As an outlook in the future, one can combine these two effects in one nanostructure, thus sufficiently improving the photoluminescence of quantum dots under two-photon excitation. For details, please read our recent articles on this subject. Thanks for your attention.